Hi everyone, welcome to today's lecture on Monday. How was your weekend? I hope you guys had fun. How was your Super Bowl experience? Yeah, I mean, we are not in class, so you got to talk to your roommate about it. But basically, what we are going to learn today is about prokaryotes, and basically, we are going to learn about bacteria and archaea or archaea. So the learning objective is as listed below. First, we will distinguish between different shapes and arrangement of bacteria. We will localize and define the functions of structures all bacteria possess and the structures that only some possess. We also summarize the processes of chemotaxis and we describe the composition and model the formation process of biofilms. And also we will also differentiate between two main types of bacterial envelopes, the envelope structure and also molecules which are used in each, and how gram stain techniques is also dependent on each. And then the pointers is available. We also talk about the model and the processes of sporulation and germination and state why. And we also talk about why endospores are a medical problem. And lastly, we will also compare and contrast archaea to bacteria and and eukaryotes. So as we've talked about earlier on the first slide, we um we are talking about the classification prokaryote. Oh no. We are talking about bacteria and archaea, but they are classified as prokaryotes, which are no nucleus or organelles, and they are mostly smaller in size. One example is as we've seen below, cyanobacteria, and basically you can see they are like they depends on photosynthesis to survive, and the major component of plankton and water dwelling. As you can see, they are extremely smaller. Today's slide is a lot, so I'll try and go through it as efficiently as possible. It will include most of video links, which you have to watch it by yourself. So here under this slide, we are going to talk about why bacteria are single cell organisms. Basically, they are just cells, or the bacteria is basically cells, and they are capable of carrying out all necessary life activity. They can be free living, which are like planktonic, and then we have bacteria that can also act as a group. So basically, like we talked about in the group, we talked about in last class, we talked of colonies. So basically, a colony is just a group, a single colony is just a group of bacteria, and we can see them. And mostly the colonies have formed biofilms in order to protect themselves or just nourish the environment. And as you can see from here on the plate, on an agar plate, you can see a single colony. And then this is a microscopic view of it, in which just a single colony have a lot of them living in it. So, what is normally the size of a bacteria? Averagely, their size ranges from 1 micrometer to 10 micrometer, and you can see from microscopes structures. And then, because they are within a very huge range, we have the ultra small bacteria, which is mostly 150 to 300 nanometers. And one example of them is the mycoplasma. And as you can see from this image, it's really small, but when you zoom in, you can clearly see their size. And then we have the ultra large group. An example is the Margarita species. And then here you can actually see them. These are actually macroscopic. You can basically see them. You can basically see them, and you can see how long they are. They're actually longer compared to what we see from here. And sometimes can also be seen with the naked eye. The macro group, which is the Theo Margarita species. And then we are going to talk about shapes of bacteria. So they actually comes in 6 different shapes. We have the coccus, basically spherical in shape, and we have the bacillus, which is rod, as you can see from here. And then we have the vibro, which is basically having a one edge curve in a rod shape, as you can see from here. And then we have the spirillum or spirillum, depending on who is saying it. Basically, as in spiral, they come in a corkscrew shape. And then we have the spirochete, which comes in very long and also twisting, as you can see from here. And then we have lastly the filamentous shapes, or basically forming of filament or branches, as you can see from here. So as much as we've known the bacteria shapes, we also have to understand their arrangement. So they come in different form of arrangement. That is the arrangement based from the coccus group. So the arrangement I'm going to talk about is from this coccus group, those who form the spherical shape. So back to this slide. So we have the diplococca, or the diplococcus, or the diplococci. Basically, they are just two individual bacteria being joined together, which is basically formed from cell division of the bacteria themselves. And then we have the tetrads, as in tetra, basically four. And we have the sacina, basically cubical packet of 8, 16 or more cells. And then we have the ones which form the irregular clusters. So as you can see, just a lot of them all clustered together. These are coming in as a staphylococci, and then we have a chain, those who actually form like a longer chain, which is the streptococci. And then we also have the other different bacterial arrangement, which include the Diplobacilli. These ones are coming from the ones who form the rod shapes. That's the bacillus. So the Diplobacilli, we have basically two rod shape. Streptobacilli, more of a longer rod shape. And we have the pacillates, which is basically like trying to attach to each other on the side. And then we have the spirola, which ones are like basically occasionally short, and they are occasionally short chains and very raw, as you can see from here. And there's actually a lot of them. There are other ones which still remain, which still rarely remains and attach even after cell division. So mostly I will have read this question and tell you guys to answer, but because there's a lot of like a longer trend today, I'll try and skip it, but try and answer these questions. You actually have access to this slide, so you can go to this slide and try and answer them. So we will then skip to discussing or trying to understand the basic bacterial cell structures. So as we can see from here, bacteria, like any other cell, have a nucleus, have a cell membrane. You can see from here, some bacteria have three layered cell membrane, others have two. We talk about that, and as you can see from here, there is also an external structure, which is sorry, which is the fimbriae. We will talk more about it, and then they also obviously have a cytoplasm. They will have a nucleus, and the list goes on and on. So the first structures we want to talk about is the external structures, which is basically the appendages, which is made up of flagella, villi, and fimbriae, which we'll talk more about it. And they also have stuff on their surface. It can be an S layer or glycocalyx. So we'll talk more about it. Ignore the other ones. We are only focusing on this one, which is the external one for the meantime. So we only pick the first one, which is the appendages. So in relation to appendages, most bacteria or some bacteria will be having external structures outside of the cell membrane itself. And there are two major groups of appendages. The first one is flagella or axofilament, as I've been shown here, and their main function is basically motility to help the bacteria move. And then we have the second group, which is fimbriae and pili. Basically, their function is to how the bacteria attach to surfaces, or it's composed of channels, which helps in the transfer of stuff in and out of the bacteria cell. So the next one, one I want to move in. So now we are going to tackle them individually. So now we are moving to flagella. So flagella, as we've said from the previous slide, is for motility and is present in some bacteria. Structurally, is made out of three hooks. As you can see from here, it's made out of three parts. First one is the hook, filament, and then the internal part, the basal body, which is basically hooked or connected into the cell body of the bacteria itself. And basically, what it does is just rotates to provide movement, and it's basically driven by a protein motive force, gingered by hydrogen ions. So, so on this slide, there's different part of the flagella. And since you guys have the slide, I wouldn't want to go deep into it, but basically there's just a lot of labeling all over. And I will entreat you guys to watch these videos. So when you click on this link, it will send you to your YouTube and you basically will learn more about it. But because of the time and what we have to learn today, I'm leaving this up to you to click on this link and then watch the video. So under flagella, we have different kind of flagella arrangement. So we have there are two groups. We have polar and non-polar. So under polar, basically we have the monotrichous, which means it's just in as we can see from here, it's just one polar or one way or one direction. And then we have the low foot trichomes, which is basically having more than one flagella, as we can see from here, still in the same direction. And then we have amphitrichos, basically having it on both direction of the bacteria. And then we have the non-polar, basically it doesn't follow any direction. And as you can see, under non-polar, we have the petri trichos, which is having the flagella all over the body of the bacteria. So now they have the flagella, which is basically helping them to move. But where are they moving to? Are they moving towards something or are they running away from something? That's basically what we are going to talk about. So on this slide, we have chemotaxis and phototaxis. So chemotaxis basically means the cellular movement of the bacteria in response to a chemical signal. And basically, it can be positive or negative. So positive chemotaxis means the bacteria is moving towards a chemoattractant. It can be nutrient, it can be chemical signal. And as you can see from here in this picture, the bacteria is basically moving towards the chemoattractant. And we have the negative, which is basically moving away from the chemoattractant. It can be phenol, it can be bile, it can be anything that is harmful to the bacteria, and it just tries to run away from it. So let's talk more about bacterial chemotaxis. So the chemotaxis basically means the movement of the bacteria itself. And then the movement comes in two forms. Either it's a run or it's a tumble. So run basically means moving in one direction or moving towards a place. And basically it's been triggered or it's been aided by the counterclockwise movement of the flagella, which is behind. So it's just like, like a tadpole or a fish swimming and it's just shaking. But this time around, it's just moving in a counterclockwise direction and it's just helping the bacteria move straight. And it can be smooth, it can be linear in direction, and it's basically towards the stimulus, which is the chemical attractant, which is attracting it towards or it's just moving away from it. And then we have the tumble, basically the flagellum reverses in direction and it can either stop or help the bacteria stop or change course. So instead of now moving counterclockwise, it's just be moving counter anti-clockwise and basically it's helping the bacteria here just move or change course or change direction. And as you can see in this image, um, under normal condition, bacteria is just moving, it can be moving in any other direction. But once there's a chemo attractant, as we can see from here, now the bacteria is basically moving towards that chemo attractant in this image. I hope it is very clear. Now, let's talk more about the part of the flagella. So we have the periplasmic flagella. Basically, it's just made up of acyl filament, and the main function is to help in motility or movement, as we've already seen previously. And it's more present in spirochetes, and as we can see, nicely arranged. And then you can, for this parts of the whole thing, you shouldn't worry much about it. But the whole idea is to get how it works. And you can also click on this link and watch a YouTube video about it. Mostly in class, I would have played it, but you guys now have the ability to do that. Gen Z world, you should do it. So here, back to what we are discussing. We have the internal flagellum, and it's mostly between the cell wall and the cytoplasmic membrane. Main function is to twist and help in flexible movement. And as we can see from here, this is just, let's say this is the bacteria, and then on the cell wall, we have these periplasmic flagella around the outside of the cell wall, enabling the bacteria to move. And now, lastly, we have fimbriae. So, fimbriae is basically present in some bacteria. It's some bacteria, and it's small, and it's bristle, looking like fibers covering all of the body, the surface of the bacteria. And as we can see in this example, you can see fimbriae all over the bacteria itself. It's mainly composed of the composition varies and can be made up of it's mostly made up of protein itself. What is the main function? As simple as ABCD, it helps the bacteria attaches to surfaces of stuff. So, for example, let's say we all have gut microbiome, right? So you have bacteria or stuff in your microbiome, and the whole goal is for them to attach into the intestines or the microvilli of your intestines to prevent them from being washed out. So next, we move to pili or pili, depending on who is pronouncing it. But basically, it's just a long, rigid, tubular structure which is made up of peeling proteins and is mostly present in gram-negative bacteria. So it's only present in gram-negative bacteria only. And the main function is to just attach or sometimes used in mating through conjugation. So conjugation basically means transfer of DNA from one cell to the other. So for example, let's say this cell is mating with this one, it's just transferring their DNA to this one, and this one transferring their DNA to that one. So a newly discovered structure which is being seen is called nanowires or nanotubes. So these are actually thin, long tubular extensions of the cytoplasmic membrane. Main function is serve as channels for amino acid and electron shuttling. So both in and out, basically in and out of the bacterial cell. And you can also click on this link and it sends you to the source and you watch more videos on it. So electron shuttling is a way to generate energy in oxygen-dependent manner, and then the bacteria can draw electrons from electron-rich surfaces such as iron-rich salts or sediment also into the bacteria itself. So it's not only one way, it's just both in and out. So here is also another practice question. You should practice this by yourself. It's not difficult, or let's say you have to work on it by yourself and then give an answer. If you can't, you can see me in class on Friday and I'll give you answers. But please give it a try and also read the stuff on Model and Connect to get more idea about it. So now we've moved from appendages, so we are now moving to surface layers. So with surface layer, just imagine you are back in the, I don't know, is it 1700s or 1800s medieval era, and you are like a knight. You're going for war, you gotta protect yourself. So basically, that's what the surface layer is. just made up of thousands of copies of single proteins linked together, and is present in some bacteria. The main function is as that provide protection, and it's only produced in a hostile environment. So if the bacteria hasn't faced any danger, it's not going to produce much of it. But when there's a danger or where they are living is very hostile, then they have to produce these. And then on the surface, under this S layer, there's also another group which is called glycocalyx. Basically, it's also present in. Oh no, what happened? There's a bit of technical glitch, so give me a minute. Let me move back to it. Okay. Yes. Now we are back in. Now we are back again. So glycocalyx is present in some bacteria and is basically made up of polysaccharides, which is sometimes you have some proteins also in there. So the function is to also protect, and sometimes it's aids in the attachment, which is basically made up of which is basically done with biofilms. And they are made up of two types. So we have the slime layer and also the capsule. So we first talk about the slime layer and we talk about the capsule. So the slime layer is actually looser in composition and could easily be washed. And the main function is to protect the bacteria from water and also nutrient loss. So as you can see, it's just slime as in slime. Let's say this is the bacteria. This is the bacteria. So it just form a bit of slime layer all around it, just preventing it from water and also nutrient loss. Then in the next one is the capsule. So the capsule is more like denser compared to the slime layer and it's actually tightly bound and is typically found around pathogenic bacteria and it protect them against phagocytosis or phagocytic white blood cells. So now instead of the first one protecting them from water, this time around what they are forming, which is the capsule, is actually protecting them from organisms attacking them. And some example is the phagocytic white blood cells. So in case you get infected with a bacteria and you are not getting cured, just know that that bacteria is busy forming capsules and it's invading your white blood cells. Yeah, so this is how it looks like. This is the bacteria cell body itself, and then it has formed the capsule or the protective covering around itself, and it's very hard now for the white blood cells to attack it. It's mostly so basically just as you can see, it just look bulkier and larger in shape. Then we also have some specialized function of this glycocalyx, which is biofilms. So now the bacteria have now evolved into we've now moved from bacteria forming external structures like flagellum and now to bacteria forming or secreting things to protect themselves. So biofilm. So biofilm is basically composed largely of polysaccharides, which is glycocalyx itself, and also they are being present. They also have extracellular DNA in proteins and sometimes enzymes. So they are simple. They can be single or multicellular species. These can be present in both single and multicellular species, and they can include more than one bacteria. So, for example, if you have one bacteria secreting biofilm, it can attract other bacteria, and they all come together and keep secreting more and more. But hey, this is a prime example of how it looks like. So, I'm sure we've seen much of these where you have this green stuff, these green bacteria forming all around a rock or something like that in water body. That's basically bacteria, a lot of bacteria forming biofilm, and it just looks slimy or it might look shiny when you see it. So it's actively present in dividing or metabolizing cells. And then, yeah, this is a prime example of, someone's teeth if you don't brush it well, and there's a lot of bacteria present forming biofilms. So biofilm is good for the bacteria for them to survive, but it's also bad for us. I mean, those who are being infected by the bacteria, because there is a clinical problem. Biofilm itself is very resistant to antimicrobial treatment. So the bacteria had formed their barrier, they have been resistant to treatment. So most of the time, it's been known that this bacteria, this antimicrobial treatment of biofilms is at least 10,000 times. Sorry, it's at least 1,000 times compared to what we've seen other microbes have. It's actually now very difficult to penetrate into this biofilm. Now, why? Because it's very difficult to degrade them. Their size is actually exclusion, and they are also present in toxic environment. It's very difficult to deal with it. They are commonly found growing on medical devices. So now you go to hospital, they have these catheters, they have to insert in you, try to deliver liquid or try to deliver drugs into you. And now you have these bacteria forming biofilms all around these catheters. And now what they do is they either cause infection or they cause damage or corrosion to the device itself. Some microbes form biofilms during infection also as well as we've talked about it. They get into your tummy or get into your blood system, your gut or your system, and they form the biofilms and then protect themselves. So now let's talk about how biofilms itself is being formed. So biofilm is being formed by when, let's say this is the surface. You have a single colony come, it attached to this surface, and then it forms a little bit of the biofilm, and then it keep forming more, and it then basically glycocalyx formation starts, and they keep forming this glycocalyx formation, and then they keep attracting because they have this biofilm present, they keep attracting more, and like basically the ecosystem keep growing where you have a complex bacteria, everybody keeps coming, joining the community, and they keep growing and growing. And that's what basically how biofilm is being formed. So we can talk of quorum sensing. Basically, is the ability of a bacteria to regulate gene expression in response to sensing of bacterial density or population, important after the cells start dividing on surface and also thereafter. That is the whole mechanism. They attach, they divide, they attract other people, and they join. So now let's practice this biofilm modeling. Um, Usually breaks down into steps. So, but this is a representation of the fluid process. This one you have to discuss with your colleagues and come by with the answer. Again, you have these slides, you have to practice this by yourself. And then the same thing also happens to this slide. You also have to practice it by yourself. So now we've discussed about the outer surfaces, we've discussed of appendages, we've discussed about what is on the surface itself. Now we are going to talk about the boundaries, basically the outer membrane, the cell wall, and the cytoplasmic membrane. So bacteria, like any other cell, obviously have a cell membrane, and then the cell envelope or the cell membrane is composed of two or three basic layers. So we have the cell envelope composed of three basic layers. So we have the cytoplasmic membrane, which is present in all bacteria. We have the cell wall, also all present in all bacteria except the mycoplasma. And then we have the outer membrane, which is present in some bacteria, which is the gram-negative bacteria. And as we go, we will understand why the gram-negative bacteria actually have the outer membrane. So let's tackle the outer membrane first, only present in gram-negative. So that is this outer membrane is the exterior to the cell wall, as we can see from here. So this is the cell wall, and then you have them like this. You have the outer membrane now present. Basically, it's just made up of a lipid bilayer embedded with a lot of proteins. The function is to help structure and then select is to help structure and help in the selective permeability and transport of proteins in and out of the bacterial cell. So this is actually an example of it. You can see in the outer membrane there, you can see like more of pores on the surface or just porous selective permeable membranes, protein structures on the membrane, as you can see from here. So on the outer membrane, there are different kind of structures on it. And what I want us to take a look at is this lipoproteins and then liposaccharides. So first, let's handle the liposaccharide and then we'll come to the lipoprotein. So basically, this is how the liposaccharides look like. They are mostly found on the outer lipid bilayer of the membrane and they are present on the outer portions. And it is basically a variation of polysaccharides and they are being detected by the immune system. And they also have an inner portion, which is the endotoxin. Basically, the endotoxin is the endotoxic shock, which is caused by the lipid protein. So this is like when they get into your body, they deliver this shock, and as you can see, someone is sick and they have been experiencing the endotoxin when the bacteria got infected in them. So now we've finished discussing the liposaccharide. Now let's move to the porine proteins, which is these ones, the pores in them. So they are all over the outer membrane and they allow relatively small molecules to penetrate into the bacteria and they are not substrate specific. For that reason, they make the outer membrane more permeable than the cytoplasmic membrane itself. Just look at think of it like the TSA at the airport where you have to get through them before you go and board your airplane. And then lastly, we move to the cell wall itself. And as the cell wall, we already know the function. It helps determines the bacterial shape. It helps in the structural support to prevent the bacterium from bursting or collapsing under osmotic pressure. Basically, think of the cell wall as a tire protecting the inner delicate tube. So as you can see, on your bicycle, you have a tube, and then you have an outer surface. That is basically the cell wall preventing the bursting of this guy, the tube itself. And it's basically the cell is comprised of peptidoglycan, except for the mycoplasma species, which doesn't have it. And they have different thickness. They have different thickness depending on whether it's a gram positive or gram negative bacteria. So now we've moved past the cell wall, and now we have to also talk of the peptidoglycan structure. So the peptidoglycan structure basically comprises of the glycan chains, which are cross-linked with short peptide fragments as cross-linked, as you can see from here. They are just connecting to each other. The peptide are just cross-linking to each other, as you can see from here. So these are all chain linked, or they look like fences crossing pattern, as you can clearly see from here. And they are having an alternating sugar glycans or an N-acetoglycan and N-acetylmuric acid. I know this structure might look complicated, but just look at it as how beautifully these sugars are being connected to each other. So basically, it looks like a fence, as you can see from here. So let's talk about the gram-positive bacteria and how their cell envelope look like. So we spend all this time spending much more about the gram-negative. So now it's time to talk about the gram-positive. So with the gram-positive, we realize the gram-negative have all these three layered cell envelope, which is like the outer and it keeps coming back to the cell membrane. But for the gram-positive, they actually look different. They have a thick peptidoglycan, which is 20 to 80 nanometers of a cell wall, very, very thick, as you can see from here. And then within that, they have a wall Tioic, tioic acid, which is WTA. It is made up of a polysaccharide attached to the cell wall. And as you can see, it's just present here. And they also have the LTA, which is polysaccharide attached to the cytoplasmic membrane. And it just passes through the cell wall itself. So both the WTA and LTA function in the cell wall to help in maintenance. And then because it's negative and it's negatively charged on the cell surface, so it helps its role in cell signaling too. So back to the gram-negative cell wall again, and as you can see, compared to the cell wall of the gram-positive, which is thicker, the gram-negative is more thinner, as you can see from here, very, very thin, which is 1 to 3 nanometers in shape. I'm sorry, nanometers in thickness, and which gives it more greater flexibility, but more sensitive to lysis. And it's having a periplasmic or space or perisma, which is present on it. And as you can see, I think this one gives a clear surface. So we have the outer membrane here, we have the peptidoglycan in place, and we also have the cell membrane, which looks really nice. So now let's do a comparison of the gram positive and the gram negative cell envelope. And as you can see, gram negative is having the outer layer being present here, and it's having, and both of them will now have a peptidoglycan, or basically a cell wall, which is present between both of them. Gram positive have a very thicker cell wall, gram negative have a very thin cell wall, and then both of them now have a cell membrane, which is present on both of them. So in 1884, Hans Christian Gram developed a method for distinguishing between these two groups of bacteria, which is the Gram stain. So he developed the Gram stain to help us tell between a Gram positive and a Gram negative bacteria. So basically, how this works is as simple as I'm going to describe it. You come in with the first reagent, which is crystal violet. Crystal violet is added to this cell in a smear, and it's basically just stain all of them in the same purple color. So looking at their cell wall, one have the gram negative, three cell layer, gram positive have two. So both this cell wall itself is being stained, and both cell walls are stained, and it's just fixed with the dye. And then next is to stain them with iodine or iodine. It depends on who is pronouncing it. This is just a stabilizer that causes the dye to basically form larger complexes in the cell wall, and then the thicker gram positive cell walls are able to more firmly trap the larger complexes than that of the gram negative. So as you can see from here, you add iodine, it's making it's making them form bigger complexes in the cell wall. Gram positive is thicker, so it can withstand that pressure. In the gram negative, there is no effect of the iodine because their cell is very thin. The next is to add alcohol. Basically, is to dissolve lipids in the outer membrane and remove the dye from the peptidoglycan layer or the cell wall only in the gram positive. So now when you add it, the crystals remain, when you add alcohol, the crystals remain in the cell wall because they actually have a thicker cell wall, so it's not easy to get all of it out. Compared to the ones which is the gram negative, which have the thinner cell wall, you're able to get the cell outer cell membrane is then weakened, and you're able to get most of the dye out. And then lastly, you stain all of them with safranin, which is the red dye. And then at the end, you will now be able to now visualize that when you have a red dye marked by violet, you will now be able to know that that is a gram negative. And when you have only the red dye and the cell is colored, that's a gram positive, because those ones have a thin cell wall, and basically we end up losing the crystal violet in them. So that is basically the mechanism in which the gram staining developed by Hans Christian Graham. That's how it works. So now we talk more about the cell wall and basically what is in gram negative and gram positive. There's another group which is basically non-typical cell walls. These cell walls lack cell membrane structure, or they basically lack the cell membrane structure of either a gram positive or gram negative bacteria. There's no peptidoglycan present. Sorry, the peptidoglycan is still present. And examples of these bacteria include the mycobacterium and the nocardia. And what are the unique structures of these non-typical cell walls? Basically, they contain unique lipids, which is mycolic acid. This is very long fatty acid chain, and they contribute to pathogenicity and acid fasting. I mean, this really look, you can clearly appreciate that this really look differently than the ones that we've seen so far. This makes them more resistant to certain chemical dyes, including the acid fasting. So let's talk about mycoplasmas. These are extremely ultra small bacteria, and most of the time, or all of the time, they have no cell walls. They really don't have any cell walls, and they are sterile stable. So no matter how much you sterilize them, they are actually stable, and they are pleomorphic in nature, and they vary in shape and size. An example is Mycoplasma pneumonia, which causes walking pneumonia. And as you can see, they really have complex shapes and sizes. As you can see, one is looking like an S shape, and now you come here, you have the long shape one, and then the sizes keep changing over and over. So their cytoplasmic membrane are stabilized by sterols and is resistant to lysis. So that's something I wanted to add to for you guys to remember too. So now let's talk about the cytoplasmic membrane. So this is present in all bacteria and is present in the interior of the cell wall and the exterior of the cytoplasm. This is a biolipid layer embedded with proteins. As we can see from here, this is where it is. What is the function? The function is basically to help structure and select permeability and also transport of proteins in and out of the bacteria. So it's very selective. So as we can see from here, it is a membrane channel embedded into cell wall. And then basically you have non-specific lipid going in there. And then obviously you have other also lipid soluble molecules also leaking through or just moving through them. So the selectivity or the permeability of this part of the cytoplasmic membrane is very selective. It just allow selective movement of water and smaller uncharged molecules, and basically just diffused. They all diffuse freely into it. And they also have special carrier mechanism that helps in the exiting of most of these molecules. As we can see, one of them is this channel here. So now let's talk about how these things work. So now we have a gram negative bacteria here, we have a gram positive bacteria. And you can also always remember the complexity of the gram negative bacteria cell membrane and how it looks like compared to the gram positive one. So up here we have a hemoglobin. We have the hemoglobin present, and we also have a heme present. So in the gram negative bacteria, these ones are diffused through these channels, and then with the help of this protein, they are then transferred throughout the whole membrane and is then delivered into the cytoplasm of the gram negative bacteria. A similar trend is then now seen in the gram positive, where hemoglobin or heme in itself is just transported through the membrane, the outer membrane of these bacteria, and is then transferred into the internal part of the inner part of the bacteria. All of these mechanisms, which we are basically these specialized transport systems, function differently, and they are being done with the help of numerous proteins, as you can appreciate on this graph. So now we've now learned about how things get in. How about how do things get out from this bacteria, or how does things get out from the bacteria itself? So basically, there are specialized transport systems with many working parts or numerous proteins, which are called secreting systems. So basically, now we've let's say we've import the bacteria have ingested or import a lot of things into the cytoplasm and they want to get rid of whether waste or any other thing. They are basically these very complex thick fragilar structures which we've learned earlier. We have flagella which are present on the membrane of the bacteria. So the main job is to, as much as helping the movement, the bacteria also uses this in secretion or discharging of metabolic products which they've already produced. So an example is this. Let's see this T3SS um, structure being formed on the membrane, and basically let's say there's a metabolic waste in here and it just basically moves through and it gets secreted outside. Example is also CTS T6SS, and basically it's also all of them does the same thing, helping the elimination of metabolic waste. Now let's shift gears and talk about antimicrobials and also the cell envelope. So, when the bacteria goes into your body, it tries to interact with your immune system and try to um, evade it and or break it down and attack you and just make you get sick. So, there is an interaction with the immune system, and it's basically the bacteria is causing a host toxicity within you. The gram-positive bacteria are more sensitive to their cell wall, and then which makes it possible for us to apply antimicrobials, basically degrading these cell walls. However, the gram-negative ones, which are more like which have a thicker membrane, are more sensitive to membrane targeting antimicrobials, but they are more resistant to many other antimicrobials due to their uh, basically their three-layered protective shield that they have on. So let me give an example. For example, penicillin kills susceptible bacteria by specially inhibiting a peptidase, which is a PPP that catalyzes the final step in the cell wall biosynthesis and basically helps in cross-linking of the peptidoglycan, and then it causes the cell death of that bacteria itself. So you can clearly appreciate how the formation or the structures of the cell membrane can be targeted using antimicrobials, and it will help in the killing of the bacteria when we get infected with them. So now this is time for you to practice. You can draw the gram-positive and the gram-negative um, bacteria envelope, and then you can label it. This one you can do by yourself, so I wouldn't go much into it. Now it's time for us to discuss the last part, which is basically the internal of the bacteria. And then we are going to discuss more of like cytoplasm, ribosome, and other part of the bacteria, like any other cell type. So, bacteria internal structures. These are present in all bacteria. And then there are a lot of them, and we are going to go through them step by step. First one is the cytoplasm. The main function of the cytoplasm it basically forms as the main site for biochemical activities, and it's about 70 to 80% of water and complex. It's made up of about 70% to 80% of water and complex mixtures of sugar and amino acids. Next, we have the ribosomes, which function as which is function as a spot for protein synthesis and is found inside the cytoplasm. Comprises of protein and ribosomal RNA. And and the next one, we have the chromosome, which is basically houses genes, basically either a single or circular strand of DNA in the cytoplasm. And basically in the cytoplasm is located Oh, wait. Hmm. Yeah, so the chromosome just helps in the basically within the chromosome is a lot of single or double-stranded DNA, and basically in the DNA contains all the coding sequences you will need for coding all the genetic makeup of the bacteria itself. So in the bacteria have its own DNA, and then they have an extra pair of DNA, which is a plasmid. So as you can see from here, this is the DNA of the bacteria, and it just have this circular extra DNA which is present in itself. This circular DNA, which is the plasmid, is separate and is being seen as circular pieces of it, and then it's heritable. And the main function is not like not it's non-essential DNA, but basically it helps in protecting of the bacteria against extra dangers that it might possess. And then it's basically used in when you come to modern cell biology, it's just used in genetic engineering, where we kind of target we take a bacteria plasmid, you kind of clone anything on it, you put it into the bacteria help duplicate your target sequence, and then you can use it for your research. So something to take from here is that the bacteria have its own DNA and have another pair of DNA, which is circular plasmid, and it's just used in protecting itself. It have all these protective traits that you ever find in the bacteria. So within the bacteria, we also have the inclusive bodies and microcompartment, and it's present in some bacteria, but not all. So let's tackle the inclusive bodies. The main function is for storage. So it serves as a storage site for nutrients during periods of abundance. And also it's enclosed in a single layered intracellular membrane. And then it also varies in size and also numbers and also content. Next is the microcompartment, and the main function is to produce protein or enzyme proximity, and is then enclosed in a protein layer. And as we can see from here, this is an example of microcompartment seen in the bacteria. Then we have the cytoskeleton. So in some bacteria, they possess this cytoskeleton-like structures, which are long polymers of proteins, and it's actually similar to the actin cytoskeleton in eukaryote. And as you can see, the main function is to help in cell shape and cell division. Next is the bacteria endospores. So when the bacteria faces danger and then they want to protect itself, most of the time some of the bacteria end up producing endospores, which is basically a mini version of themselves, and is being formed to protect themselves so that they can kind of revive themselves later on. So the main function is to survive hostile environment, and it's mostly found in gram-positive bacteria or soil-dwelling organisms. They have two phases of life, which is vegetative cell and then the endospore phase, which are they are just metabolically inactive. The endospores are metabolically inactive, and basically the function is to preserve the DNA and protect the bacteria from extinction. So endospores itself is just resistance to a lot. They are heat resistance. They can stay in extreme cold or weather conditions, and they have cortex and endospores coat protection, as you can see. So this is the core endosperm itself. Endospore itself. And then in there, after the core endospore, they will have a protective cortex all around it. And then they also have an endospore coat. So you can clearly see why they are actually resistant to heat and all other extreme conditions. But there is a problem with endospore because they are very difficult to kill, and then very difficult to eradicate. For that reason, they are just easy to spread because they are mostly smaller in size too. And as you can appreciate, someone had been infected by endospores. and you can clearly see that their lives don't look good. So they contribute to the spread of medically problematic bacteria. So some bacteria that produce endospores include the Bacillus anthrax, which results in anthrax. And then the list goes on and on and on. They are resistant to boiling water, soaps, disinfectants, you just name it. So now let's talk about how these endospores form and what is the whole cycle of an endospore. So now let's just focus on here. This is a healthy bacteria living its life. Everything seems fine. And then it just realized maybe it's have less nutrient or there's hunger or there's danger and it start developing, it start programming itself to develop the endospore. So here now it develops or makes a duplicate of its own self or the whole genetic makeup. And then it start modulating its membrane to create an inner endospore. So basically here it creates the early endospore itself, and then it starts forming the cortex or the first phase of the protective coat around the endospore, and then eventually it gives up on itself or give up on itself, and then it dies and basically leaving the endospore. And now the endospore can then move around, resist all heat and everything until later it's ready to kind of give birth again. And then, sorry, not give birth again, to be revived again. And then the bacteria can then be produced back into its normal self. These are links to animations explaining how this whole thing happened. Please click on it and use it. So this is also for you to practice. Please take some time and practice it. So now we've talked more about bacteria and everything. So now it's time for us to move to Archaea or Archaea. So the Archaea, they really look like bacteria, but they are not bacteria. They have similar size, around 1 to 10 micrometers. They are single, and they have just circular chromosome. They do not have organelles. They do not have organelles compared to bacteria as we've seen so far and talked much about it. They share cell structures with bacteria, and they also have their 70S ribosomal chromosome. Sorry. They have their 70S ribosomal chromosome classified, and they are all ubiquitously expressed, and they are also part of the microbiome. So now let's talk about some unique structures, which first one is the ribosomal RNA sequence. They really have their own unique ribosomal RNA sequence, and it's not found in it's not found in bacteria, and they are more genetically related to eukaryotes. So you can realize why they are quite different from bacteria. And then they also have their ribosomal 70S, but they are structurally distinct, and they have protein synthesis, which is similar to eukaryota or eukaryote. And then they also have a cell membrane and basic cell membrane lipid. Basically, these are long chain branch hydrocarbons. They are not fatty acid as the one we saw in the bacteria, or polysaccharides as the one we saw in the bacteria. And they are with either linkage. They are with ether linkage instead of the ester linkage that we also saw earlier on. And as you can appreciate, as seen here in this figure, they are actually presenting with ether linkage as we can see from them. 5. There is no peptidoglycan in their cell wall. Basically, it's just made up of polysaccharide or protein instead. And they also have flagella. This time around, a solid fibra-like structure, and it's also ATP driven, as we can see from here. Appreciate how it looks like. Most of them are most of them are extremophiles. Basically, they can live in heat, salt, high, very salty places. extreme heat, pH, just hydrostatic pressure, they can really withstand all of this. But note that not all of them are extremophiles. Not all Archaea are extremophiles. And then lastly, they are non-pathogenic. It means they can't cause disease. So as much as they have all these extreme wonderful structures, they can cause disease. If you want to know much about these two, please click on this link. It will send you to the YouTube where you learn much more about it. So lastly, for you to practice this, go to Connect, go and look for bacteria and archive review worksheet. Please work on it and practice and get more idea about it. Yeah, sorry, today's lecture is extremely long. It's been over 1 hour. I tried as much as I can to cut it short. So if I went too fast, please play the video over and over again. Try to get it. If not, shoot me an email and I'll explain more to you. Also, try and go to Connect, read more about it, and do the worksheet and practice more. Also, try and do the assignment on Moodle. And I will send you another link on Wednesday, and we shall meet in person tomorrow. Sorry, yeah, tomorrow, Tuesday, for the first in lab. Remember, bring your lab coat and get everything ready. Tomorrow is the hay experiment, so you can come to class with your grass for the hay inoculation. Come to class with your grass for the hay inoculation. Remember your lab coat. And then I'll send you another video on Wednesday. And then Friday, we will meet in person. Have a nice day and sorry for the long talk. Bye.